All right, so we're going to hop on to a new chapter then, chapter 5, structure and function of large biological molecules. Now, before we continue, I did say this in lab, I'm probably going to refer to all of those large molecule groups as macromolecules. All right, strictly speaking, remember, only 3 of them are macromolecules, and very strictly speaking, 3 of them are polymers. The lipids aren't. But I'm going to kind of talk about them generically as big molecules. All right, is everybody okay with that? Okay. All right, this is an awesome picture. Look at that.
Is that from that one
Bodyworks.
Yeah.
It's from Bodyworks. It takes you a while to actually look at that and really understand what's going on, doesn't it?
Oh,
Yeah. So there's a Bodyworks exhibit in Vegas that I'm going to go look at. I'm going to take the kids as well. It'll be awesome. So they've done these exhibits for elephants and camels and horses, and it's amazing how they do it. But so you can literally see a human head and neck and the insides of it, right? All of your biomass is essentially made up of 4 major chemical groups. Everything you see there is essentially made up of 4 major chemical groups. Okay? All right. So then we're going to look at, in chapter 5, the biology of these 4 classes of molecules that you're made up of. So, all living things are made up of these 4 groups of biological molecules. We've got our carbohydrates. Give me another group. Proteins, good.
Lipids.
What was that?
Lipids.
Yeah.
And then your acids.
And nucleic acids. Okay, they're our 4 groups. Okay, so I want to define macromolecules. We're going to define a few terms. Macromolecules are large molecules, and they are very complex. Right? As the name implies, macro means big, and a molecule is what?
Macro small.
Macro, right, macro's big.
Okay.
A molecule is what?
Small elements. Complex. Compound.
Give me the definition of a molecule.
Oh, it's 2 atoms together.
2 or more atoms bonded together, right? That's a molecule. So, these are very, very, very big molecules. There are many, 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 many atoms, sometimes thousands of atoms in these molecules, all right? And they can be extremely complex. Now, these very large biological molecules have got unique properties, and those properties arise from their structures. The structures are literally how the atoms are arranged in 3-dimensional space in that molecule. Okay? Now, the carbohydrates and the nucleic acids and the proteins, they are macromolecules. They can be enormous, and they are all polymers. Now, I'll give you a formal definition of polymer in a moment. There are 3 of the 4 classes of biological molecules. The other ones are the lipids. Now, the lipids can be big, but they're not as big as the others. And the lipids are not polymers, even though they are themselves made up of smaller building blocks. Okay, it's a subtle distinction, but it's an important one to understand. Carbohydrates, nucleic acids, and proteins are enormous, and they are all made up of smaller building blocks joined together. Many, 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 many of these building blocks. The lipids are smaller. They're made up of smaller parts, but the smaller parts they're made up of, we don't refer to them as monomers because they can often be quite different. And not all lipids are made up of smaller parts joined together. As we talk about these groups, that will become clear. But I'm sometimes going to refer to them all as macromolecules. Okay? But strictly speaking, the lipids aren't. I don't think I'll ever refer to the lipids as polymers. If I do, that's a mistake. And you can correct me. Okay? And you get a donut as a reward. Not really. All right, so this is what one of those enormous molecules might look like. In fact, this is the three-dimensional representation of a protein. Now, what you can't see in that are the atoms necessarily. You can see a few of them. See a few of those atoms there? But this long chain, which sometimes is a coil and sometimes these sheets going backwards and forwards and sometimes seems to have no rhyme or reason, that's strings of amino acids, which are themselves made of atoms. Really big, really complex molecules. All right, so let's define polymers and monomers then. Each cell in your body has got thousands of different macromolecules, all performing different functions and having different roles. But that huge variety of different macromolecules can all be built up from a relatively small set of building blocks, or monomers. Okay? So here's a good analogy for you. Think about this. We've got only 26 letters in the alphabet. And from those 26 letters, not many, just a few building blocks really, how many words do you think we can make? How many official words are there in the English language? It's very hard to count how many official words there are, but what's the rough count? How many words in the English language?
I'm not sure.
Anyone want to guess? 100,000?
30 million.
It's just over 1 million.
What?
Topped 1 million.
Oh, really?
But there's a whole bunch of words that are very debatable and, they're very controversial about whether they should be considered a word and so on. But if you go have a look, it's a hard count, but they reckon we're just over 1 million. 1 million words in the English language. And we can make an enormous diversity of big words from just 26 little letters. Okay? The same is true of these complex biological molecules. We can make thousands of different molecules from just a small number of building blocks. And guess when you learned this? As soon as you could play with Legos. Okay? Think about it. A box of Legos doesn't contain very many different parts, does it? Right? But you can make an enormous variety of structures from just a few different Lego pieces, as long as you've got enough of each of them. Right? Yeah, make something like that. Now, if you were ever into this show, that would mean something to you, but if you're not, you won't know what that spaceship is. It was a good show, wasn't it? Can't believe they took it off. Oh.
The movie's real good.
Yeah, movie was good. Anyway, point is, look, you can make a crazy number of big things from a small variety of building blocks. Same concept applies to these big biological molecules.
So the monomers are the big ones, and the polymers are the
Monomers are the small ones, the small building blocks
that make the polymers.
that we link together to make big molecules, the polymers. Yep. All right, so a polymer then is a large molecule that's made up of many similar building blocks that are covalently bonded together. That's what a polymer is. It's a really big molecule made of lots of little similar building blocks that are covalently bonded together. Now, when I say made of many building blocks, many monomers, how many are we talking, you think? Yeah, it could be, sometimes it's not a lot. Sometimes it's maybe 20 or 30, depending on what the molecule is. But sometimes it could be hundreds or thousands of these small building blocks joined together to make these massive molecules.
The polymers, in a sense, are usually big molecules to begin with. So, I mean, whether it's, like you said, it's hundreds of thousands or whatever. The fact that
The monomers can be reasonably big.
Okay.
I mean, that's all relative, right? But, all right. Okay, the repeating units, the building blocks of the polymers are called monomers. Mono means what? One. Poly means many. Makes sense, doesn't it? Think of the word, what's that white, very light substance that you get that almost every electronics is packed in?
Oh, that styrofoamy stuff?
Yeah, styrofoam. I know you guys say styrofoam, but you should actually say
Polystyrene.
polystyrene. Good. Polystyrene. Styrene is actually the monomer. Let's join styrene molecules together and we get polystyrene. Plastic bags are polyethylene, right? Lots of ethylene molecules all joined together to get polyethylene. All right? All right. Okay. So remember, 3 of the 4 classes of macromolecules are polymers. Which are the 3 that are polymers? Good. And which of the big molecules is not a polymer? Good.
But lipids aren't necessarily monomers.
Lipids themselves are not monomers, and they are not made up of monomers. Okay? So polymer and monomer only really apply to those 3 groups. Okay? Yeah, I think we'll stick with that. Lipids are made up of smaller building blocks, but not many repeating similar ones. All right. Okay, so you're all okay with polymer and monomer. All right, so concept number 3 then is dehydration and hydrolysis. So we're going to look at how these monomers are joined together to form the polymers. And then we're going to look at how the monomers are broken apart so we can disassemble a polymer into its constituent monomers. All right? Okay. All right, so let's have a look at this dehydration and hydrolysis, these 2 reactions. But first, these things in your body, many chemical reactions in your body don't mysteriously occur. I mean, many of them do. They happen spontaneously. But many reactions in your body are facilitated by enzymes. Now, enzymes themselves are specialized macromolecules. In fact, they're typically proteins. And the enzymes are able to speed up reactions that would have occurred anyway. They just make them go really fast. And it's the enzymes that join together the monomers to make polymers. And it's the enzymes that break down the polymers into the constituent monomers. So there are enzymes that are doing this. Okay? I'm not going to show you the enzymes most of the time, but you've got to understand that when 2 monomers join together, there's an enzyme that facilitates that. And when monomers break apart, there's an enzyme which drives that process too. So, let's talk about a dehydration reaction then. A dehydration reaction occurs when 2 monomers bond together. They join with the formation of a covalent bond, and when they join together, forming that covalent bond, a water molecule is lost. Or I could say a water molecule is formed, and so you can think about water as being lost when the 2 monomers join, and that's where it gets its name, dehydration. You're removing water, it's like you're dehydrating. And it's a dehydration reaction where we join monomers together. That's how we build up polymers. So we build them up with dehydration reactions. I'll show you some graphics in a movie in just a moment so you can sort of visualize the process. But is everybody okay with what a dehydration reaction is? It's kind of what we did in lab. So we also need to disassemble polymers, break them apart into their constituent monomers, and that process is called hydrolysis. Now, if you were to break apart the word hydrolysis, you could break it apart into hydro and lysis. What does hydro refer to?
Water.
Or water, yeah. And what does lysis or lytic refer to?
Cutting.
Breaking or splitting.
Breaking.
Hydrolysis is literally water splitting, splitting water. So when we undergo hydrolysis reactions, it's kind of the reverse of a dehydration reaction, but we break apart monomers, and each monomer that we break off from another one requires a water molecule. We have to break a water molecule. And this is how we break down polymers, through hydrolysis reactions. Where do you think your body does an awful lot of breaking down of polymers? Your digestive system, right, good. All right, so I'm going to summarize a dehydration reaction. Okay, I'll give you some steps, and then we'll look at this diagram. So through the progress of a dehydration reaction then, one monomer is covalently bonded to another, that we know. And that results in the formation of a water molecule. To form that water molecule, a hydrogen is removed from one monomer, and a hydroxyl group is lost from another. The hydrogen and the hydroxyl groups join together. That forms the water molecule, and that makes the bonds available so that the monomers can bond. So let's have a look at this diagram then. I'll use the cursor. This number 1 and this ball represents a monomer, just a generic monomer. Could be any one for any of the different groups. And it's already joined to a second monomer, which is already joined to a third monomer. But we're going to add a fourth monomer, this one over here. Now this monomer happens to have a hydrogen free at the end, and this monomer has a hydroxyl group on one end of it and a hydrogen free at the other end of it. And there's a whole bunch of other atoms in the middle which are specific to that monomer. Okay? In order to join this monomer to this growing chain, then we need to remove the hydrogen and that hydroxyl group. They join together to form the water, and that lets this number 3 join with this number 4 to forge a covalent bond and link it together. Now you can see that this monomer here has an available hydrogen, so we can just stick another monomer on if we want. And we can keep adding monomers as long as we need until we've made the molecule that we want. Okay? Yeah.
Would that just continue every time you add a monomer to
Yep.
the existing? It just brings out another water molecule?
That's it. So when number 1 was joined to number 2, see this bond? That released a water molecule. When 3 was joined to 2, that released a water molecule. When we join 3 to 4, that releases a water molecule. So when we formed this growing polymer with 4 monomers, be careful at your answer, how many water molecules did we make? 3. Good. Okay. Everyone okay with that? All right, now let's look at hydrolysis. You can almost think about hydrolysis as being the reverse of dehydration. Okay? First, the covalent bond linking two monomers is broken. Okay? So let's focus on this bond between these number 3 and 4 monomers. We're going to break this bond. Now when we break that bond, we've got something on number 3 and something on number 4 which is unbound, right, when we broke the bond. And we need to add a hydrogen to one and a hydroxyl group to the other. To do that, we split a water molecule. A water molecule gets split into a hydrogen and a hydroxyl group. The hydrogen joins one monomer, like it did just there. The hydroxyl group joins the other monomer. And we can continue to break down that big polymer into its component monomers simply by cleaving those bonds at the ends with a hydrolysis reaction. Good. So how many water molecules does it take to fully break down that 4 monomer polymer? 3. Yeah. So dehydration reactions actually produce a lot of metabolic water inside your body. And that's an important source of water. Okay? In lab, we talked about kangaroo rats, right? Which don't need to drink free water. They produce most of the water they need from metabolic water produced by making polymers. They've also got a special kidney, a very well adapted kidney, which produces super concentrated urine so they don't lose very much. So hydrolysis reactions, where do they predominantly occur in your body, you think? Where? Where in your body would you get a lot of hydrolysis reactions? Breaking down polymers into monomers. You said it earlier. Digestive system. Think about all the food you eat. It's all made of polymers. Well, as long as you eat real food. Donuts, I don't think contain any polymers, right? Well, actually they do. They contain a lot of starch, but that's about it. So think about the food you eat. It contains polymers. You've got to break those polymers down. You've got to break them down into their constituent monomers. One reason is so that you can absorb them into your blood. So your digestive process is primarily about breaking down those polymers into their component monomers, which are small molecules, small enough for you to absorb into your blood. They move around your blood system. They move into cells, tissues and organs. Maybe you modify them a bit more, but then in your tissues and organs, you can join those monomers back together to form the polymers that you need. Okay? All right. So this graphic then just summarizes dehydration and hydrolysis. Is everybody okay with those two? It doesn't matter whether you're talking about carbohydrates, proteins, or nucleic acids. It's the same generic reaction. And even with lipids, with many lipids, when we join them together from their small parts, we also undergo dehydration and hydrolysis reactions. Okay? All right. Good? Okay. It's always better to watch the movie, isn't it?
Most biological molecules are very long and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
So that big purple thing and that beige
Yeah.
thing are enzymes?
Enzymes,
Okay, that's cool.
which are themselves molecules, usually proteins, massive ones. Now, did you notice how the enzymes perfectly fit the monomer? The monomer just fit properly. Yeah?
Yeah.
So that's important, that fit.
They do fit, like.
They do. They physically fit. Not quite, but almost like jigsaw puzzles fit together. In fact, what happens is the fit is not quite perfect, and then when it comes in, they kind of
Instead of just
properly fit.
bond, they actually come together.
They do. Yep. And there are enzymes that do this.
Pretty cool.
Isn't that cool? All right. Oh, what happens if we want to make lots of one particular polymer? We might need lots of that enzyme, right? What happens if we wanted to stop making the polymer? What could we do to that enzyme?
We could put it somewhere else.
Put it somewhere else? It's hard to move molecules around sometimes, ones that are that big.
Break it down.
What about change its shape a little bit? So it doesn't work quite as well.
Wait, what changes shape?
The enzyme changes shape, it might not function the same.
And so how do you change it? Yeah.
How can we change the shape?
pH.
Maybe change the pH. Yep.
Then
Okay.
you said wouldn't it react and act differently? If you change the shape, it wouldn't be the same?
Yeah.
It can change the shape, and it doesn't do its job the same. Right?
What would be like a real life example
Of what?
of like why would you want to change that?
Change the shape of the enzyme.
Yeah, why would you?
I'll give you an example. In your cells, you've got some very highly destructive enzymes, which are designed to, for example, when your white blood cells engulf a bacterium, yep, it's got to do something with that bacteria. It's got to destroy it. Yeah, kill it. And what it does do is, if here's your white blood cell, it's just engulf a bacteria. Let's just say that's a bacterium. And it keeps it in a little pocket, right? Literally a little cavity inside the cell. There's the bacteria. I've exaggerated the size of the bacterium compared to the cell, but anyway. So your white blood cells produce these highly destructive enzymes, all right? And they house them in these little vesicles, these little bubbles, called lysosomes. They're lysosomal enzymes. So now there's a whole bunch of chemicals, and it floods the lysosomal enzymes in there. And those lysosomal enzymes break down and destroy all of the polymers in the bacterial cell. They literally just chop all the polymers up. But if this is a lysosome full of those highly destructive enzymes, what's to stop those enzymes breaking down the white blood cell? What you can do, just imagine you keep the pH inside the lysosomes at pH 5. That tweaks the shape of the enzymes and they don't work properly.
So the white blood cell can have an effect on the pH of the enzyme?
Yes, it can just flood it with hydrogen ions, right?
So it keeps it from eating the white blood
That's
cell.
right, keeps it inactive.
So like with hydrogen peroxide, how does that affect?
Oh, that's different.
Okay, my mom always said not to use too much because it can start to kill the good stuff.
It's just hydrogen peroxide is a very reactive substance.
Okay, never mind.
But it's good at killing bacteria. So pH 5, they're inactive. Let's release them in with the bacteria. pH changes, now they're active.
So the blood has its own control system over those.
The blood has buffers which keep the pH very strictly regulated.
Buffers.
Yeah?
Those guys.
All right. Is it coming together a little bit?
Yeah.
Yes. I'm enjoying it.
All right, so concept number 4. What we're going to do now is we're going to look at all 4 of those groups one by one, and we're going to look at their structure and their biology. Okay? So we're going to start with the carbohydrates. Right now, every morning when I walk into lecture, I'm carbohydrate starved.
Yeah.
My blood sugar's through the just tanked out. I'm not thinking properly. Stumble a little bit. It's not because I'm drunk. All right, but anyway, no, I've started to eat super low carbs just to dump a little bit of weight and so on and so forth. But these are the molecules that contain an awful lot of calories in your diet, right? Refined carbohydrates these days form an incredibly large proportion of your diet. But now you get an idea of what you're actually eating. Yep, we're going to look at their structure and their function. You can see chemically what you're eating. So when we talk about carbohydrates, we're talking about polymers that are made of sugars and the sugars themselves. Is that a surprise to anybody? Carbohydrates. The starch that's in potatoes, the starch that's in pasta, the flour that's in bread, even the cell walls of the plant cells in grains are all polymers of sugars. Just glucose molecules joined to one another. And glucose itself is a carbohydrate. So that picture there sort of quite nicely represents quite a span of carbohydrates. This is what many of the sugars look like. White powders. And then when we polymerize them, we get a whole variety of different carbohydrates. All right, so carbohydrates are sugars and polymers are sugars. See that potato? Mm-hmm. When you look at it, what do you see? What should you see? When you look at that potato, you should see this, a big pile of sugar.
Oh.
It's literally what it is. With a bit more water, or a bit less water in the case of the starch. Okay, so carbohydrate molecules then are polysaccharides, big carbohydrates are polysaccharides, and they're polymers made up of sugar monomers. So pretty much all what we talk about as complex carbohydrates are carbohydrate macromolecules, which are polysaccharides, and they're polymers made up of sugar monomers. All right, so let's have a look then at monosaccharides. Monosaccharides typically have a chemical formula that has a carbon to hydrogen to oxygen ratio of 1 to 2 to 1. All right? So monosaccharides typically have molecular formulas that are multiples of CH2O, in which case the carbon to hydrogen to oxygen ratios are a 1 to 2 to 1. In fact, if you think about the word carbohydrate, if you break it down, literally what does it mean? What does the carbo refer to? Carbon, good. And the hydrate refers to? Water. It literally means carbon of water. Okay? So, carbon to hydrogen to oxygen in that ratio. That's what monosaccharides typically have.
Always that same ratio?
Typically, yeah. In monosaccharides. I can't think of an exception. There almost always are. All right? Kate, what's the chemical formula for glucose? Write it down. What's the ratio of carbons to hydrogens to oxygens? 1 to 2 to 1. Good.
Wow.
Yeah, wow. So let's have a look at some examples of some monosaccharides. One. Glucose is probably the most common. C6H12O6. Okay, it's probably the most common monosaccharide. What do we know about glucose? Go on, just tell me some things real quick.
It's a sugar.
It is sugar. Good. It looks like that in the picture. What else do you know about it?
It serves as a form of energy.
It does. It's one of the main energy molecules of your cells. What else do you know about it?
It's great. I love it.
Sorry? It can, and it's a complex process going from sort of carbohydrate to fat, but yep. Does it taste, is it super sweet?
Yes.
I like it.
It's fairly sweet. There are other sugars that are much sweeter. All right. What else do you know about it?
Helps control your insulin.
Ooh, no, it doesn't.
No.
Insulin helps control glucose levels, though, the other way around.
It corrodes glycogen.
Think plants.
I think plants.
Good. Plants produce it by photosynthesis. Yep.
I thought that was just assumed.
Okay. Here's another example of a monosaccharide. Fructose.
What?
Fructose. No, you don't say fructose. You say fructose. Go on, say fructose. You're not swearing. It's okay. Fructose. Sorry?
It says.
Oh my goodness, look at that. The chemical form is exactly the same.
Is that intentional?
It is.
Okay.
It's not a trick.
Okay.
Chemical form is itself. Same. It's also C6H12O6. How can you have two different substances with the same chemical
Are they
formula?
different shapes?
It's got the same number of carbons, hydrogens, and oxygens. They're just arranged slightly differently. I'll show you how they're arranged. All right? Okay, so fructose. Oh my goodness. The amount we hear these days about high fructose corn syrup. Yep. Avoid it. Is fructose a bad substance? It's fruit sugar. Plants produce it. It's found in fruit.
It's like acid. Not buying it in moderate amounts. It's only too much that's bad.
The problem seems to be, and some of the reports are a little conflicting, it's hard to figure out the truth, is with high fructose corn syrup, it's a man-made substance, right? We take corn, which has a lot of starch in it, we chemically modify it and break it down to make what should be equal amounts of glucose and fructose joined together. Okay? That doesn't quite happen. In the chemical process, there's a little bit more fructose than glucose. All right? And there's also probably some byproducts, which are neither. And the problem is, I think, is those byproducts, but also your body is not familiar with this unfamiliar ratio of this man-made chemically produced sugar. All right? So, fructose itself, though, is perfectly natural. So is glucose. Now, it doesn't mean to say just because it's natural, it's good for you, right? Scopolamine is a natural substance. You have too much of it, you don't make it. Okay?
But that's true of anything.
Sorry?
But that's true of anything.
Yeah. Galactose is another example of a monosaccharide, which we're going to talk about. And look at that. It's also got the same chemical formula, C6H12O6. Isn't biology convenient? You've got 3 different substances and they've all got the same formula. You don't have to remember different formulas. Yeah, designed just for you. All right, so most of them look like that white powder. So, let's have a look at the structure of glucose then. We can draw the structure of glucose like this. 1, 2, 3, 4, 5, 6 carbons. If you count the hydrogens, there are 12 of them. And if you count the oxygens, there are 6 of them. This is the chemical structure of glucose. Now I want you to pay attention up there to the orientation of that oxygen, which is double bonded to that last carbon, and then the hydroxyl group on that carbon there. Now let's take galactose, same chemical formula, but we're going to just simply flip this around. Whoop, flip it around. So now it's on the opposite side to the molecule as that oxygen. Yep, same formula, different structure. Structure determines function. 2 different sugars. Now let's look at fructose, and that's quite different still, isn't it? There, we've got the double bonded oxygen, not the terminal carbon, but the carbon just before the terminal carbon. Same chemical formulas, different structures. Structure gives functions, so they've got different functions. Good? All right, let's wrap it up there then. Good job.